Today we'll be talking about chemolithoautotrophs and the extreme environments they're found in. First, let's define what a chemolithoautotroph is. So the prefix chemo means chemical, signifying that its energy source is from chemicals. Litho describes the chemicals used for energy as reduced inorganic compounds like nitrogen, sulfur, iron 2, and manganese 2. Hydrogen sulfide is the most common compound used, but metal sulfides, elemental sulfur, and thiosulfate are also utilized. Auto signifies the microorganism's carbon source is inorganic. These include carbon dioxide and bicarbonate. The term lithotroph was created from the Greek terms lithos meaning rock and troph meaning consumer, together meaning eaters of rock. Pretty cool, right? Many of these organisms fall into the domains bacteria and archaea. Common examples include various nitrifying bacteria, prokaryotes from genus Theobacillus, and ones from genus Beggiatoa. The process of chemolithotrophy is defined as the oxidation of inorganic chemicals to generate energy via oxidative phosphorylation. This process is similar to aerobic and anaerobic respiration, but instead the electron donor is an inorganic compound. The electrons are passed off to carriers within the electron transport chain, creating a proton motive force that is used to generate ATP with the help of ATP synthase. Now that I've explained the basics, let's dive into some of the environments they're found in.
The first extreme environment is deep-sea hydrothermal vents. Here, the movement of magma through the seafloor causes hydrothermal convection, and hot water full of minerals flows through the vents. These vents can reach temperatures of up to 400 degrees Celsius. Ouch. The vent fluid is full of reduced minerals that act as electron donors. This is why the entire food web in vent communities is based on chemoautotrophy. There are three mechanisms of carbon and energy transfer to the food web in vent communities. The first is the symbiotic relationship between vent bacteria and tube worms. These worms have no digestive system, so they depend on sulfur-oxidizing chemoautotrophs living inside of them for nutrition. The hemoglobin in the worm's blood binds hydrogen sulfide, and it transports it to the chemoautotrophs. The bacteria oxidizes the hydrogen sulfide and fixes carbon dioxide into organic compounds. The bacteria benefit from the nutrients in the worm's body, and the tube worm uses the microbial fixed organic carbon to grow. Now that's a win-win situation. Another mechanism is microbial gardening. This is when bacterial cultures are maintained by mussels and other invertebrates on specialized appendages like gills and tentacles. The invertebrates periodically harvest and eat microbes, but leave enough to start a new crop. I guess anyone can have a green thumb, even without fingers. The last mechanism of carbon transfer to higher trophic levels is deep-sea organisms feeding directly on chemoautotrophic producers. Crabs, fish, and even other microorganisms feed directly on the chemoautotrophs. These examples all show how important chemoautotrophs are in the ecosystem, and without them, these unique communities could not exist.
Acid mine drainage systems are another environment in which these organisms are found. These are anthropogenic environments that contain pyrite deposits that include various heavy metals such as silver, gold, copper, zinc, and lead. These minerals also make up various sulfide mineral compounds. Mining practices expose these sulfide minerals to air and water, generating various biological acids. Excess water can create acids with a pH lower than 2, and when met with a toxic metal mixture, they create acid mine drainage, or AMD for short. These conditions of extreme acidity allow for many specialized microorganisms to populate these kinds of environments. Like the hydrothermal vent community, the AMD community is self-sustaining and based on chemoautrophy. The overall diversity of these systems is not very extensive due to the unique pH levels and sulfur-based minerals and metals present there.
Another example of an extreme environment in which chemolithoautotrophs are found is the desert carbonate cave. Desert carbonate caves typically lack phototrophic primary production due to the lack of sunlight in these types of environments. Most caves receive fixed organic carbon from drip water or streams, which promotes the growth of calcium carbonate formations called speleothems. However, in desert carbonate caves, input from drip water or streams is rare, which results in highly oligotrophic conditions. A primary example of this type of environment is the Kartchner Caverns, located in Arizona. The presence of Chemolithotrophy was found in this environment through examining a clone library. Two types of Chemolithoautotrophs were found, one that obtains energy from nitrogen oxidation and the other that obtains energy from iron oxidation. Chemolithoautotrophs are also capable of significantly contributing towards primary production through ammonia oxidation. It has been determined through thorough research that these microbial communities have genetically adapted to the low nutrient conditions of the desert carbonate cave. Desert carbonate caves are unique in that they have a very high concentration of calcium in their environment. When calcium concentration is too high, calcium toxicity can occur and cause DNA strand breakage. To counteract this issue, Chemolithoautotrophs have an overrepresentation of DNA repair enzymes in their genome. Because of this, they are able to tolerate or even thrive in the oligotrophic conditions of desert carbonate caves.